biography briefly from your LinkedIn profile?
Okay. Yeah, that sounds good.
Typically, I find this like they are still up to date. So
yeah, it's reasonably up to date.
Okay, good. So today we have Dr. Zach Bent from 10x Genomics, and he's been there for about 6 years at 10x, and he's the director of consumables and product development. But recently, I think he's the director of spatial portfolio development. And previously, he was a postdoc in the systems biology research at the Sandia National Labs for about 4.5 years, where he worked on RNA-seq analysis of pathogens and how it affects the host transcripts. And then prior to that, he was a graduate researcher and graduate student at UC Davis, also in California, for about 5 to 6 years. And he did his also BA degree at Cornell in microbiology. And I think today we're going to be hearing about the technologies that 10x has been developing. And recently also, as we were chatting at the beginning, they acquired a couple of new identities, and we're going to hear about those new identities and new capabilities of spatial omics as well. Without further ado, I think I'd like to turn to Zach.
Great, thank you for that introduction, and thanks for inviting me here today. I'm definitely appreciative of the opportunity to speak to this group and tell you guys about some of the stuff we've been working on in the last year or two with the Visium portfolio. So I titled my talk today, Spatial Resolve Whole Transcriptome Data with High Resolution and Morphological Context Using Visium. So I probably don't have to explain to this group why spatial analysis is important, but it's because location matters. Really what we're looking at here is gene expression and spatial organization together, rather than doing dissociation and doing single cell. So spatial profiling offers some significant improvements there. Gives you biological information while retaining the organization of the tissue environment. You don't have that disassociation bias, so I think oftentimes we see tissue disassociation. You might lose certain cell types, or you might bias towards certain cell types. If you just slice the tissue and look at it directly, you don't have any of that to worry about. And you can identify cells in the context of their heterogeneous tissues. And so the Visium spatial gene expression solution provides spatial whole transcriptome analysis that allows us to look at gene expression and maintain that spatial organization that's so important. I've always liked this picture here, thinking about different RNA-seq methods. As you heard, I've been working on RNA-seq for about 10 years now. Started very early on with bulk RNA-seq, what you see here is Legos on the left. Essentially just a mismatch of all your Legos in a single block, and you kind of get an average. More recently, we've seen huge advances in single-cell RNA-seq, where you can now see the individual cells that make up a tissue. And then you add that to the spatial transcriptomics, and now you can see not only those individual cells, but where they are in space. And that allows you to really study that functional tissue and the whole tissue together. And so we look at single cell analysis is really very, very complementary to spatial analysis. And these two things together can really provide that whole functional tissue look. So what is the Visium spatial gene expression solution? At a very high level, you can use this to obtain whole transcriptome, high throughput gene expression analysis of intact tissue sections from a wide variety of sample types. And I'll have a slide in a little bit that tells you some of the sample types we've validated in-house, although it's certainly not in the limited list there. There's a lot of things you can do with this that we haven't even looked at yet. Allows you to unravel biological architecture in normal and diseased tissues, discover new tissue biomarkers, and we've got an interesting paper I'll highlight a little bit looking at that in the brain. It allows you, you can integrate this easily into your laboratory already, so you don't need to buy any specialized equipment as long as you have a cryostat and a microscope. We provide the rest of the reagents you need to make this happen. So it's really a pretty easy end-to-end workflow. And we've streamlined this to the point that you can do this from section to library in essentially a single working day. So the main features, we offer whole transcriptome, whole tissue. This is spatially resolved your total mRNA with all the morphological context using poly-A capture. We think this has relatively high cell resolution. This is 1 to 10 cells on average per spot, which of course depends a bit on the tissue you're looking at. Some tissues have higher cell density than others, but each one of these spots is about 55 microns in diameter, and it can contain about 1 to 10 cells in that spot. As I said, there's no tissue disassociation, so you don't have that disassociation bias that you can see in single cell. More recently, we launched the ability to do co-detection of proteins. So this is through immunofluorescence. We also have streamlined data analysis tools. So this is the Space Ranger pipeline as well as the Loop browser that allows you to visualize your data. It's efficient. We have a one-day workflow. It's pretty easy to use. We do naive user training all the time. People pick it up very, very quickly. And it's very straightforward to run. And it's kitted, ready to use, and all the reagents are right there. And again, no specialized equipment is needed to run the Visium platform. So what does a Visium slide look like? If you look here, you have your Visium slide. You guys can see my cursor, right?
Yes, we do see.
Yes.
Okay, good. So you can see the 4 capture areas on these slides. Each one of these capture areas, this total area around is about 8 millimeters squared. The 6.5 millimeters squared here refers specifically to these black dots, which are the barcoded spots. And then these red spots are these fiducial markers that allow us to do automatic image registration. And so within these, the barcoded region here, we have about 5,000 barcoded spots in the 6.5 millimeter square area. Each one of these spots is about 55 microns in size with a pitch of about 100 microns center to center. And for those of you who are familiar with our single cell 3' product, this oligo should look very familiar to you. It's essentially the same. It's partial true-seq read 1 with a 16-base spatial barcode, a 12-base UMI, and a 30-base poly-DT sequence. So now we've split the workflow. Now there's kind of 2 complementary workflows for Visium. We have an H&E workflow and an immunofluorescence workflow. And these really only differ in basically these steps right here, as well as imaging, obviously, if you're going to do bright field versus fluorescence. But essentially, it's pretty straightforward. You take your tissue sections, you place them on the slide, you do fixation, and you do your staining, be that H&E or immunofluorescence. You do your imaging, and then you can do some on-slide cDNA preparation. Once the cDNA is made, you can take that off-slide into standard PCR tubes and create your library using fairly standard library construction procedure. That goes onto an Illumina sequencer. You can sequence that, get your data out the back end, process that with our Space Ranger or third-party software, and then you can visualize this using Loop or again third-party software if that's what you want to do. So most of you are probably aware that this technology actually came from a SciLife spinout, the company Spatial Transcriptomics. We acquired Spatial Transcriptomics kind of the fall of 2018. And the first thing we wanted to do is really to drastically improve that product line, make it a true 10x product. And so we focused on a couple different areas. We wanted to first increase the resolution, second increase sensitivity, and third streamline that workflow. And so you can see here a mouse brain using the original Spatial Transcriptomics. There were about 1,000 spots in the same area that we put 5,000 spots. And those spots were significantly bigger, about 100 microns with a 200 micron center to center pitch. And so we're able to drastically improve the resolution here, and you can see much better structure because of it. We were also able to significantly improve the sensitivity. In most cases, we see 3 to 5 or 6 times more genes per spot, and up to 10 times more UMIs per spot. So we're really able to increase the sensitivity by about an order of magnitude across most tissue types at that same sequencing depth. And we're able to do all this while simultaneously shortening the workflow from a 3-day workflow to a 1-day workflow. So what are we actually selling here? We're selling the two basic kit types. We're selling the tissue optimization kit. This is a kit that allows you to find out the optimal permeabilization conditions for your sample. It comes in just one variety right now. It's 4 samples, so it gives you 4 slides. Each slide gives you the opportunity to optimize one tissue. And then we have the spatial gene expression kit, which is really your real experiment. This is what you're actually interested in doing. And this comes in a 1 slide or a 4 slide kit for 4 16 reactions respectively. So what does the Visium workflow look like? The first step you want to do if you're new to Visium is you need to do your tissue optimization. Every tissue is a bit unique in its permeabilization time. So we use pepsin to permeabilize these tissues, and depending on the tissue architecture, how fibrous the tissue is, how fatty the tissue is, you might notice very different times for permeabilization. But essentially, we designed this tissue optimization slide that has essentially just a lawn of these oligos. So no barcoded information, but a lot of the same oligo configuration is on the gene expression slide. And so in this case, you section your tissues, you place each tissue section onto one of these capture areas. You do your staining and imaging like you normally would, H&E staining, bright field imaging. Then you do your permeabilization time course, and you're going to do a different time for each one of these capture areas so that you can go from 3 to 60 minutes depending on the tissue type. During that permeabilization step, the RNA is able to sort of leak out of the cell and land on the slide. Once that RNA is captured by the poly-DT, you're able to reverse transcribe. And during your reverse transcription, you incorporate fluorophore labeled nucleotides into your growing cDNA strand so that the growing cDNA is actually fluorescently labeled. And you can see basically an imprint of the tissue in fluorescence after you remove that tissue and do your imaging. So what does that actually look like? This is an example of a mouse brain and the time course that we run. You'll see a lot of mouse brains in this talk. It's a really nice tissue to work with. It's relatively easy to permeabilize, and it has very nice structure that's very obvious to see spatially. And so what you would do here is you would take your tissue sections, you have a negative control where you don't permeabilize, and then you do your permeabilization time course. In this case, we went from 3 to 30 minutes, as well as adding an RNA control. And this is just total RNA that you place onto the slide to show that your reverse transcription is working. And what you're looking for here is you're looking for that optimal permeabilization condition where you see the brightest image with the least amount of diffusion. So you can see at 3 minutes, this is pretty dim, 6 minutes is a bit brighter, but 12 minutes is pretty bright. And you don't see much of an increase in brightness from 18 to 30 minutes. However, if you were to zoom in, by the time you get to 30 minutes, you might see a little bit of blurring of these lines. And that's because these transcripts are starting to diffuse away. And so in this case, you would choose 12 minutes as your optimal permeabilization time for the mouse brain, because this gives you the brightest image that has the least amount of diffusion. However, we've done studies in-house to show that this is actually a pretty broad range. So anywhere between 12 to 24 minutes essentially will give you the same or very similar sequencing results. So it's not anything you really have to obsess over. You don't have to spend hours looking at each image. It just gives you a good ballpark of where you want to start. And I do want to point out that it's really important that you do this. Even if we say that 12 minutes works for mouse brain, your mouse brain, your lab conditions, your pipetting technique might vary a little bit. And that's why it's important to do this on every step. I'd also point out that different tissue sections, so if you're looking at disease versus healthy, your disease tissue type might have a different permeabilization time than your healthy tissue type. I think you can easily imagine if you have a tumor that might be in a specific tissue, the structure of that tumor is probably very different from the structure of the healthy tissue that's next to it. And so permeabilization times can even vary within tissues when you have disease and healthy tissue. However, once you've kind of found that tissue for that permeabilization time for the tissue that you're interested in, you're pretty much good to go. You don't need to redo this every time. You can take that same 12 minutes here that we found, and in your lab, in your mouse brains, 12 minutes is probably almost always going to work. So we think of this as an upfront thing, not something that you have to do all the time. This is a list of tissues that we validated during development. This isn't an inclusive list necessarily. This is kind of representative. You can see the full list here at this website. We've looked at a variety of healthy and diseased human tissues as well as healthy and diseased mouse tissues. Internally, we've also looked at rat. We've looked at zebrafish. We've looked at a lot of different organisms and found that it works pretty well across most tissue types and most organisms we looked at. We've even seen people coming in, customers asking for ideas on how to do it with plants. The original ST technology worked really well with plants, and we've seen some at least initial anecdotal evidence that the Visium technology would work with plants as well. So really, it's broadly applicable to a lot of different model systems. All right, so now that you've done your tissue optimization, you want to get to your real experiment. You want to look at your gene expression spatially. And so this starts basically the same way. In this case, there's only 4 capture areas per slide. But you section your tissues, you place them on the slide, you do your staining and imaging, and that can be either H&E staining or immunofluorescence staining. You image using bright field or fluorescence as appropriate. You use permeabilization based on that optimal permeabilization time that you found in the previous section. And then in this case, once you capture that cDNA, you reverse transcribe it into cDNA. The RNA is reverse transcribed into cDNA. In this case, there aren't fluorophore labeled nucleotides, they're just regular nucleotides. So you create your first strand of cDNA that's attached to the slide. You can create a second strand on the slide, and then you denature that second strand off the slide and transfer that into a standard PCR tube, and you can continue doing your cDNA amplification and library construction in a standard PCR tube. And that library construction is kind of that classic fragmentation, end repair, A-tailing, adapter ligation, and sample index PCR that's common to essentially all of our products. So if you've done any of our single cell products, you're probably pretty familiar with this type of library construction. So we support sequencing this on the NextSeq, HiSeq, and NovaSeq. It actually works on all Illumina sequencers, but you just don't get enough data on the smaller options. So we do have tons of data on the iSeq, MySeq, MiniSeq. It all works. It's, if you're just running a QC or trying to understand the library quality, that's a good way to go. But for your experiment and looking at your gene expression, you really need to use a sequencer that gives you a bit more depth. In terms of the reconfiguration, we do use dual sample indexing with 10 base sample indices. Your read 1 should be 28 bases to incorporate your 16 base barcode and 12 base UMI. And so our recommendations, and this is now true across our product lines, including our single cell, is 28, 10, 10, 90. Although that 90, depending on your sequencing configuration, can vary. There's sort of a minimal difference between like 75 and 120, but 90 is definitely the optimal. And we also recommend a minimum of 50,000 read pairs per tissue covered spot. So, if your tissue is covering the whole capture area, that's 250 million reads, but most tissues don't cover that whole capture area. And so you can actually get by with quite a bit less than that 250 million that you would have if your tissue covered everything. So once you have the sequencing data, you want to analyze that, and we've developed the Space Ranger pipeline as well as the Loop browser to do that. These are programs that are freely available on our support site. Now the Space Ranger allows for automated data and image processing. It does clustering analysis and based on gene expression. So it almost treats these spots as if they were cells. You can get these spatially naive T-SNEs. It actually also generates actual QC statistics. And once you've gone through that, you can look at the output in loop. And this is interactive data visualization exploration, and allows you to easily export and share your results and generate publishing quality images directly from the browser. This is an example here of a breast cancer tissue that we sequence in-house, looking at it through the Loop browser. What you can see here is a lot of different features. You can see these 12 different clusters that were identified using the spatially naive clustering. You can adjust the spot opacity here and see more what your tissue looks like in the background. You can also look by cluster and see what are the genes that are most upregulated in cluster 1. Here you can see IGCL is one of the top upregulated genes here compared to these other tissues. And these numbers here in parentheses are the number of spots that are incorporated into that particular cluster. And so there's a lot of different ways you can visualize your data in Loop, and it's really kind of fun to play around with and look at what these different structures are doing. And that's another thing. You can just compare regions of interest. So if you didn't want to rely on the spatially naive clustering and you were just interested in 2 specific areas of a tissue, you can highlight those and do differential gene expression between those 2 areas. In this case, looking at area 1 versus area 2, which were sort of just arbitrarily chosen. But you actually see there's a differential gene expression here, and you can see this gene is significantly upregulated in cluster 2 compared to cluster 1, or cluster 1 compared to cluster 2. And so you can basically choose what you want to analyze, although the whole tissue is there and available for analysis. We can now zoom in and do even more dynamic spot selection. So this is another mouse brain. This was immunofluorescent stained, and the graphic here just shows the ability to go in and you can highlight each of the spots that are associated with the hippocampus. And so you can go in and highlight just the spots that you're interested in, and then find out what genes are being highly expressed in the hippocampus, or what genes aren't being expressed, and just look at the total gene list and find out gene expression levels for any gene you happen to be interested in this particular region that you're highlighting. So really, Visium gives you kind of 2 types of data that you can overlay. First type is your imaging data. So every Visium output has an image of your tissue. So you can see here on the left, this is a classic H&E stain of a mouse brain. On here on the right, this is that spatially naive clustering that I talked about. This is just a T-SNE, almost pretending like the spots are cells. And so you can have these clusters that you generate without the spatial information, and then you have your spatial information in the tissue, and you can overlay those on top of each other and see how these clusters map spatially. And you can see that they do map kind of how you would expect, where these specific regions are identified by specific clusters. So what are some of the applications of this technology? So there's a lot of interest in this. I mean, I think obviously this group is very familiar and aware of the spatial field. But there are a lot of papers coming out, and it's going up and up over time, looking at either the Visium technology or the original spatial transcriptomics publications. So you can see going back to 2016 when the original kind of stall science paper was published, out to 2020, where last year there were 25 publications in kind of top tier journals using either the original spatial transcriptomics or the Visium technology. And there's actually a pretty healthy pipeline right now looking at 10x Visium preprint. So this is just Visium. We released this, actually it was only about 11 months ago we released the Visium. And there were a couple preprints in 2019 within a few months of us launching, out to 26, 27 so far this year in 2020. And these are all pretty exciting papers that we're really looking forward to see published. So one aspect of Visium, we think, spatial, and probably again this group doesn't need to be reminded, but it has its applications in pretty much all areas of biology. I think a lot of times you think of spatial and you might focus on oncology, and obviously it's very powerful in oncology, focusing on tumor microenvironments, tumor heterogeneity, as well as neuroscience, as the brain is a very difficult organ to study. It doesn't disassociate nicely. You might have to use single nuclei rather than single cells. So this gives you an opportunity to look at those large cells that have that interesting axon dendrite structure. But we also have a lot of application in immunology. You can look for immune cell infiltration. In developmental biology, you can look at embryo development really nicely on these slides as well. And finally, in pathology, you can just add significantly more data to your slide sections that you would have taken anyway. And so you can look at all gene expression rather than just some genes or a very, very subset of genes that you look at using a fish-based technology. So this is an example of a tissue we ran in house. This is a breast cancer tissue, and if you look on the left here, you can see the H&E image. One of the outputs of Space Ranger is this total UMI count, and so you can see within this tissue there are regions of very high gene expression where you're seeing greater than 20,000 reads or 20,000 UMIs per spot. Whereas there's also regions of very low gene expression where you see less than 4,000 UMIs per spot. And it just shows you kind of the heterogeneity within this tissue. You can take this to the next step and you can look at that spatially naive clustering overlaid on top of the image. So you can see that these definitely forms these unique clusters that seem to correlate well with the structure of this tissue. You can take this another way. You can have a pathologist annotate this for you. So in this case, we ask a pathologist to take a look at this, tell us what was in there. This whole area here that's kind of highlighted by this green line is invasive carcinoma, whereas the blue is immune cells, the red is fibrous tissue, and there's a couple of spots here, here, and here that is this ductal carcinoma in situ. And what you can do with the Loop browser is you can take that pathologist annotation, import that over, and then overlay and you kind of manually highlight what cells or what spots belong to which section. And what's pretty cool, and what I really like looking at and when I see this is, okay, cool. So you have the pathologist annotation, and that tells you, okay, this whole area is one thing. It's an invasive carcinoma. But if you go back a slide, you can see it's not one thing. It's very unique because there's different clusters. So there's cluster 5, cluster 1, all these different things within this one region that's been annotated simply as cancer. And it really shows the tissue heterogeneity, the tumor heterogeneity that you would expect to see. And so just because a pathologist says it's only cancer doesn't mean that that's only one type of cancer. It could be a lot of different interesting things happening within that one region. So a recent paper, the University of Tokyo, this is a preprint, was able to kind of look at the same breast cancer. In this case, they were looking at this GATA3 GATA3 mutation. And what was cool here is that the GATA3 mutation that they were interested in happens to be at the 3' end. So this is a point mutation that they're able to pick up and map spatially using this technology. So in this case, you can see kind of this, stuff that's staying dark here in the H&E was, determined to be cancer. And there were 3 clusters that correlated to this cancer, what they call 1, 2, and 3 here, as well as 4 clusters that went to the microenvironment, that are outside of these regions here that were labeled as cancer. When you look at the regions that are, determined to be cancer, and you look for this GATA3A mutation, I think it's probably a bit hard for you guys to see, but in green here, these are sort of the wild type, without the mutation, whereas these red spots that are sort of spatially distributed specifically to kind of clusters 1 and 2, you do see that point mutation in 46 of these spots. And so you do see kind of the spatial distribution of a point mutation, which is pretty cool to see. And these violin plots down here are just representative of these kind of three groups in genes that are most highly expressed. So I think A here was this epithelial mesenchymal transition, B is angiogenesis, and C was the estrogen response. So you can see, kind of global gene expression here of these genes. And then, that's correlated the violin plot, comparing the mutation to the wild type. Another really cool application out of the Lieber Institute is looking at the human prefrontal cortex. So this is a well-studied tissue type. It's been known for a long time that the cortical layers, so 6 layers are formed here. And what's cool is you could easily pick this up using the Visium technology, and it correlates extremely well to what you'd expect from the Allen Brain Atlas. And so what you can see here, layer 5 is known to have this gene PCP4 highly expressed. You can see it here from the Allen Brain Institute, and you can see it here in Visium, showing up exactly as you'd expect. And same thing with CCK, that's indicative of layers 2, 3, 5, and 6. Again, you can see high expression layers 2, 3, 5, and 6 here. And so what they found was that you could see the classical markers of the cortical layers using the Visium technology. But the cool thing that they did was they were able to go a step further. So what are the classical markers? We already sort of know that, but they were able to find previously unknown markers as well. So for example, in layer 1, they were able to see high expression of this gene AQP4, and similarly in layer 5, layer 2, and layer 6, they were able to find these previously, the genes were obviously known, but they weren't known to be markers of that particular cortical layer. And so you can use this technology to find additional information and novel findings beyond just finding what you'd expect to find in the human cortex. One of the most exciting papers, using spatial transcriptomics technology came out last year. This was a science paper looking at molecular pathology of ALS. And so mice that you give this SOD1 mutation will develop a disease that mimics ALS through the course of their lifetime. And basically, they took spinal columns of these mice and looked at, different gene signatures, and maybe able to find, this AIF1, which is a marker of microglia, GFAP, which is a marker of astrocytes, and they're able to compare the wild type to the SOD1 mutant at days, 70 and 100, and basically see that you have a massive increase, essentially, from day 70 to day 100 in the SOD1 mutant, but not the wild type. And same thing with astrocytes. So you're seeing, a change, spatially resolved change of the architecture of the spinal column here in these ALS mice. So you're able to find basically 31 what they call co-expression modules, which, I guess we could think about, as kind of T-SNE clusters in a way. And, module 8 here had 1,300 genes, whereas module 11 had, only 37 genes that made up that module. But what you can see here is kind of two different things. So in module 8, you can see the wild type mouse from day 30 to day 120, didn't really see much difference in gene expression patterns. But the genes in this module were significantly upregulated and, at the periphery of the spinal column at days 100 and 120 in the SOD1 mutant. And you see the opposite in the module 11 genes, where you see a downregulation of expression of those genes over the course of that 120 days. And again, that's very spatially defined. They were able to take that a step further and look at this in humans. So these are post-mortem spinal cord samples from ALS patients. And they're able to see, again, these co-expression modules where genes are spatially regulated, either up or down based on whether it was lumbar onset or bulbar onset. And similarly here, looking at the proximal or distal regions of the spinal column in the lumbar or bulbar onset. And especially here, you can see pretty dramatically in the bulbar onset of the distal region, a huge increase in gene expression of this gene ACHE, which has been linked very tightly to neuromuscular defects in ALS. So this was a really exciting paper that really demonstrates the power of this spatial transcriptomics technology. The other thing that we're seeing a lot of is the combination of single-cell gene expression with spatial gene expression. These are two very powerful and complementary technologies. So this paper here on the left, this is a Nature Biotech paper that was published recently. They were able to annotate kind of precise cellular composition of these distinct regions and look for spatially restricted enrichment of these different cell types in pancreatic cancer cells. And so they were able to find in these pancreatic cancer biopsies that there were definitely regions that had co-localization of inflammatory fibroblasts and cancer cells that were expressing these stress response gene modules. And either technology alone probably wouldn't have been able to get you that level of resolution. Similarly, the paper on the right is a bioRxiv paper looking at acute kidney injury as a result of sepsis. And again, they were able to show, if you see this image here, this is kind of your standard output in Space Ranger. You're looking at the spatially naive clustering. You can see these unique clusters. Running single cell analysis, you can see the single cell clusters. But when you integrate both those data types on top of each other, you can actually find distinct cell types within these various clusters and really see the evolution of sepsis in the kidney at a cellular and molecular level. So these are both really kind of interesting papers that came out recently combining single cell and spatial technologies. So this spring, we were able to release Visium with immunofluorescence. So this, initially, the spatial transcriptomics workflow, as well as our original Visium workflow, only allowed for H&E staining. But we wanted to open up the ability to co-detect proteins, so we developed the immunofluorescence ability. This is an example of that. So this is, again, your mouse brain. This is stained with new N antibody. Then you can look at the gene expression here and see RB-FOX3, which encodes new N. You see its gene expression highly upregulated here in the hippocampus. You can overlay these images, see that they overlay nicely, and you can even take that further and overlay total UMIs on top of that. So you can get these multiple layers of information now using immunofluorescence. So you can see protein expression, gene expression, as well as total transcriptome information, all in the same experiment. So I think we touched on this a bit earlier, but essentially, you can choose either H&E or immunofluorescence now. Really, the workflow is exactly the same, except for your staining step. And once you get past your imaging, there's no change to the protocol at all. So if you can do one, you can do the other. And you can get that protein information if that's what you're interested in. We also released a new version of Space Ranger and Loop that allows this to happen, and you can actually layer up to 6 different colors in the same gene expression workflow. And so you can look at potentially different stains. This could be DAPI, or you could look at like I said, up to 6 different colored fluorophores attached to antibodies, and layer all this data on top of each other. So this is an example looking at human breast cancer. So we're looking in this case at CD3 staining. So this, you can see that there's definitely regions with kind of high CD3 markers. If you look at the kind of just immunofluorescence plus the whole transcriptome, these are total UMI counts. That doesn't necessarily correlate very well to your CD3 markers, but it wouldn't necessarily anyway. But what's interesting is when you start looking at the spatial native clustering, you overlay that, you see these distinct clusters that overlay very nicely. So if you remember over here, this overlays pretty nicely with this cluster 4 here in red. And when you find out what genes are most upregulated in cluster 4, you see that they're all fairly related to immune markers that you'd expect in CD3 positive cells. And so this was a good validation of what we were looking at. You can obviously take this a step further. You can look at actual CD3 counts. So in this case, go away. That's kind of annoying. You can see that it correlates pretty nicely, the CD3 expression, and as well as other immune markers like IgH, IgL, very nicely with the fluorescent antibody staining. And so you can see that you can, overlay all kinds of gene expression data on top of this immunofluorescent data from protein co-detection. So this is something that's coming out in the next couple of weeks. We'll be releasing the Visium targeted gene expression. This is essentially based on the single cell targeted gene expression workflow that we released a few months ago. It's essentially exactly the same, some minor protocol differences that took a little bit to work out. But now that we have those protocol differences worked out, essentially you take your final library. You can take up to 8 final libraries created using either the H&E or immunofluorescence workflow, combine them together, capture them with these biotintylated probes, sequence them, and do your data analysis like you would before. So we have 4 pre-designed panels for you. We have a human pan-cancer panel. This has the genes from about 33 different cancer types, including key biomarkers, pathways, and cellular processes. We have the human immunology panel that covers innate, adaptive immunity, inflammation, and immuno-oncology. We have the human gene signature panel that looks at disease and drug targets. And when we launch the spatial target, we'll also be releasing the human neuroscience panel. So this will be a new panel that will come out in the coming weeks that covers neural development, neurogenesis, neurodegenerative diseases, and neuro-oncology applications. And you can see for any of these panels the full list of genes that are in there at our website. So what's really cool about the targeting is that you don't need as much sequencing, and you can really focus on what you're actually interested in. So this image on the left is whole transcriptome sequencing at 50,000 reads per spot, like we recommend. This particular tissue covers basically all 5,000 spots, so this would require 250 million reads to sequence this at the depth that you'd want. However, by doing your targeting, you're able to bring that 50,000 reads down to 5,000 reads per spot and see essentially the same clustering with a really nice 0.99 R squared value here. And so this correlates extremely well, and you get all this information if you're interested in these particular genes at a much lower sequencing depth. You can do a similar thing with cancer. So this is a pathologist annotation, probably from the same block we looked at earlier. You have this whole region that's invasive carcinoma. You can go in and look at the pan-cancer panel and figure out which genes are breast cancer associated. So this incorporates 196 genes in that particular panel. And you can see that these genes are very highly expressed within this region that's been annotated as invasive carcinoma. And you can take that a step further and look at this classic breast cancer marker ERBB2 and see very high gene expression again in this region that's been identified as carcinoma. All at significantly decreased sequencing depth from what you would have required before. So for the last part of my talk, I want to focus on things that we're really excited about. These are the things that are coming soon. Things that we're working on internally that we hope to release shortly. So one thing that we'll release fairly shortly, probably first part of November, is the Visium Gateway. And this is essentially a new slide type that allows you to have only 2 tissue sections per slide. And so it'll give you kind of a taste of Visium. You can check it out, make sure it works on your tissues before you make the investment to buy these larger kits. Or if you only have 2 tissue sections that you want to look at and you don't want to go use a slide that has 4 tissue sections, this is another good thing for you to use. It's got all the same, basically it's exactly the same as the normal Visium slides. You get all the same benefits. There's just 2 sections instead of 4. What I'm most excited to tell you about, though, is our work on spatial gene expression on FFPE samples. So this is our Visium for FFPE, and this is coming in the first half of 2021. And it gives you whole transcriptome analysis of FFPE tissues. Obviously, FFPE is an extremely important thing to be looking at. There's hundreds of millions, potentially billions of FFPE blocks out there. And the problem with FFPE is it's great for preserving the tissue, but it's horrible for maintaining the integrity of RNA. And so it's really challenging to get a really nice RNA, gene expression out of an FFPE sample. And so at 10x, we were actively developing a novel non-poly A-based solution for the whole transcriptome analysis. And it's got, basically going to have all the same key features of the Visium. However, it's going to be fully optimized for use with FFPE. So it's an unbiased whole transcriptome analysis, full tissue coverage, the same high resolution that you get with the current Visium platform. It's going to be compatible with a variety of histological stains. And like all of our products, you'll have all the kitted reagents and software that will be ready to use. So this is actually, you guys are lucky, you're the very first group to see this. This is some preliminary data we have looking at mouse brains again. In this case, you can see a whole transcriptome mouse brain. You get really nice clustering that'd be very similar to what you'd see with fresh frozen. All this is from an FFPE block. And recently, again, out of SciLife, they ran kind of this protocol that came out, Sevilla Campa paper, where they used kind of standard Visium technology, this poly-A capture, and they were able to show that it does work. You do get a reasonable amount of genes and UMIs. However, we're able to increase this by at least six-fold using this new technology that we're developing, and get really, really nice sensitivity. On the UMI side, even at 30 or 40,000 reads, we're not even all that close to saturation. It just keeps going up, and so there's we think that this is going to be a highly sensitive way to look at the whole transcriptome. We also looked at this in slightly older tissue samples. So this is a triple positive breast cancer sample, 2 years old in FFPE. You can see kind of your classic hematoxylin staining. The blue regions there are likely to be the cancerous regions. You can get a really nice distinct clustering with this spatially naive T-SNE that overlays very nicely with the structure that you see here in the hematoxylin stain. And you can see very good gene expression of the 3 markers of triple positive breast cancer here within these sections. And so we think that this is going to really work out well. We've even got data from tissues that are older than 2 years in FFPE. So this is something we're really excited about, and we hope to be telling you more about this in the coming months. As we talked about at the beginning, we're also really excited to add a third kind of complementary platform to 10x in our in situ platform. And this is going to come about because of the recent acquisition of Cortana and ReadCore. And this is going to give us high-plex gene expression with subcellular resolution and this microscopy rather than NGS-based readout. And we think that this will be a really complementary way to look at your data. So you could imagine using all 3 of these technologies in your experiments. And in fact, there's actually a paper, this ASP paper from last year out of SciLife that didn't do that. In fact, they looked, they created basically a cell atlas of the developing human heart using spatial transcriptomics, single cell transcriptomics, and these in situ techniques, and showing the complementarity of all these techniques. And so this was a really cool paper. And even more recently, the Cortana technology made the cover of Cell, comparing, looking at the spatial transcriptomics as well as in situ with the Cortana approach. So again, emphasizing the complementarity of these product lines. So we're real excited to see what we can do with our in situ, and hopefully we'll be getting out cool products shortly with that as well. So hopefully I've convinced you all that the Visium product line offers whole transcriptome and protein co-detection. It's very sensitive, gives you great resolution, You can have large tissue coverage, the whole tissue section. We provide this to you with kitted, ready-to-use reagents that don't need any kind of expensive capital equipment to go along with it. It's a streamlined workflow that comes along with easy-to-use and powerful software. And it's coupled to 10x Genomics world-class support team to help you through all of your problems. So with that, thank you for your time. I'll be happy to take any questions. And I guess a quick plug here, we're hiring. So if anybody's interested in working on any of the technologies we discussed today or single cell or any of the other neat stuff we're working at 10x, please check out our career website. We've got quite a few jobs available right now. So that, again, I thank you, and I'll be happy to take any questions.
Thank you for this terrific talk. Any questions? I think we have one question that's on the chat. Should I read it, or you can read it, Zach?
If you could read it, that'd be great.
Okay. Really nice talk from Caleb Stolzfus. I think the spatial clustering is very powerful. Do you have ways to interrogate these regions in depth beyond region composition, like distance to region type, or how much regions interact with or border each other? This could tell for tumor which immune-rich regions are bordering tumor regions.
Yeah, that's a great question. I mean, I think we're still at fairly early stages with what's possible with the analysis. And, that's definitely something that we're working on internally. And, we see papers coming out, bioRxiv and other places that address that. But it's not something we currently have available. But yeah, you're right. That'd be really, really neat if we could get there.
So I was impressed, actually, a quick question here, with your permeabilization thinking. I think that's a nice way of looking at like optimization, right? Currently in our cell culture protocol, we just say, oh, okay, keep it this long, keep it this much, right? We don't necessarily optimize per tissue in general. And would you also consider the same thing for a fixation process? Because I remember one of my students got a PhD qual question, how are you gonna preserve the most amount of proteins or other stuff in the cell by optimizing fixation chemistry, right?
Mm-hmm. Yeah, I mean, that's a really good question. I mean, because we do see that, just because something's FFPE or, fixed, it doesn't mean it's the same protocol everywhere. And so optimizing those protocols is probably pretty important. We haven't looked a ton into optimizing the fixation. We did with the Visium, looking at, methanol fixation versus, PFA fixation. And that preserves the RNA much better. But in terms of, fixation times, other fixation conditions, we haven't done a ton of work there. But it's an interesting point. I think, maybe we should. And maybe, it'd be great if we could, as a community kind of land on a single place that, all kind of spatial analyses could start from.
That would be great.
Yeah, that's a great point, actually, Ahmet. I think it's hard to change the clinical pathology practice, but maybe we should get it started. And if we can tell people what's the best practice to do your FFPE, your formalin fixation, that might be good for the future of the field. And but for now, I think the banked tissues for PE, I think the variability is huge. But if we can should do something like the permeabilization optimization for those samples, that's probably the short-term solution.
Mm-hmm. Well, we talk about precision medicine. How about precision reagents, right? I think that would be cool. I saw this in the context of like organoid culture development, right? Like you put additional stimulants and all these things, but not in the case of like our basic protocols, really. Yeah, that was impressive. Even though thinking about permeabilization, I really like that idea, yeah.
Yeah, we found that it's critical. For some tissues, you can actually permeabilize for up to an hour. and other tissues take a couple minutes. So it's really, you don't want to permeabilize everything for an hour because you start to get diffusion.
It's great.
Yeah, so I do have a few questions, but I would like to have the attendees ask first. I think if you want to unmute yourself and go ahead and ask. I see a couple of folks already kind of show up your face. Go ahead and ask.
Hello, this is Xiaoqing, and thank you for your great talk. So I have maybe a couple questions, if time allowed, about the procedure. If I want to work, like I want to do the spatial omics in your company. So first is, I'm still not quite sure like the procedure. So they have like different steps, like tissue prepare, staining, if I want to stain the immunofluorescence, and then permeabilization. So I need to finish like all of this step and then ship my sample to your company, or how it work, I'm not quite sure. Because afterwards have the cDNA synthesis and amplification or library construction.
I mean, we have documentation about all that on our support website.
Sure.
So, rather than going through step by step, I'd recommend you look at the support site and see, very detailed explanations of all those steps.
Sure. Okay. That would be great. So you have the document online in your your company website.
Yeah, if you go to the support page of 10X and look up the spatial section, you'll be able to find any of those documents, different demonstrated protocols, the user guides. The user guides especially will have the details of everything that you're asking for.
Sure. So also include like the design, the panel design, because some of the
So the panels are in a different location, but they're also on the support site. They're in the targeted section, I believe.
Sure. Okay, that would be good. I will look at that document. Yeah, because maybe very soon I want to start do this work. Yeah.
Cool. Well, excited to see you join up.
Sure. Thank you.
Yeah, I have some kind of biology questions. I remember, I think in one of the previous seminar presentations, I think that's last week, I think Mike talked about the tumor microenvironment. Actually, the highly compromised tumors show a better response. That's quite a big surprise. So you have the T cells in a tumor completely mixed up with the tumor cells. Actually, those patients respond much worse. But I think the imaging, either kind of MIBI or Codex, they can nicely identify the cell types, but they cannot tell you what the cells are doing, the full transcriptional or functional sort of profile. But so in this case, it would be great. So if you can use genome scale, transcriptome scale technology like a Visium to look at those kind of compromised tumors versus not highly mixed, and see, even though you have T cells there, but they're probably very different T cells.
Yeah, I agree. I mean, hopefully you should be able to see, markers of exhaustion or different, T cell states as well, whether that is, surrounding the tumor or infiltrating into the tumor.
Yeah, my thought is you see the different T cell subsets, but even the same type of T cell subtype may do very different things that cannot be captured by limited number of markers. That really, yeah, I think need a genome scale profiling to help out.
Mm-hmm. Yeah, no, I think the whole transcriptome approach will definitely give you that information, at least hopefully. I think we'll see what our customers are able to do, but that's one of the reasons we developed this protocol.
Yeah, I think another question is your single cell data integration is super cool. So we do that as well. It's super, I think, informative always. But when we look at our data, for example, the entire mouse embryo, and then we compare to the spatial pixels, we integrate them together. I think the spatial data comes from just one slice of the entire mouse embryo that does not cover all different cell types. So have you guys noticed that? Is there any ideas of how you do data integration? You can sort of annotate all possible cell types, or once you miss, maybe you can computationally infer.
Yeah, that's a really good point. I mean, you do need the thicker section. It could be a couple microns away from where you're taking that tissue for your Visium. So you could have additional cell types that are present there that aren't in your section, or vice versa. So one thing you can do is you can look at other data sets that are out there, single cell data sets, and compare those and kind of get like an average. And we've seen people do that as a way to kind of get around that. But you're right, in some cases, you're just going to be missing cell type just based on location. So you could definitely think about inferring those cell types based on, that other, data sets that are out there.
Hi, maybe a quick question from my side, if that's okay.
Sure.
Cool. So you talked about like 3 different technologies like spatial, Visium, now the new ones, Cortana and ReadCore. So what are your thoughts on, let's say, respective sensitivities of these different technologies in terms of detecting transcripts?
That's a great question. I mean, I think, we're still a ways off from understanding what our in situ project will look like. Hopefully it'll be pretty sensitive. I mean, the goal of in situ is really kind of that single molecule resolution. In terms of the difference between single cell 3' and say our Visium platform, the Visium platform is a bit less sensitive than single cell. It's hard to quantify exactly what that is because it's dependent on tissue and cell type. But I could definitely tell you that the single cell 3' product is maybe a few percent to 10% more sensitive in terms of its ability to turn an RNA molecule into a sequence library molecule as compared to Visium.
Right, thanks.
Great. Any other questions?
Yeah, I think the last slide is most important, Zach. So I wonder if you want to kind of further tell people what types of positions your guys are looking for, maybe. Yeah,
I mean, we're looking, if you check out our website, we're looking for positions, across our entire organization. So, sales, support, within the science, we're looking for computational biologists, software engineers, molecular biologists, chemists, microfluidics experts, imaging experts. We're looking across the board. There's, probably over 100 open positions right now as we're growing really fast. And, we'd be excited to have, people in this group, apply.
Exciting. Then let's thank to Zach again for this terrific talk. And I think it's clapping time. And with this, I think we are concluding our seminar series 3. Yeah, clappings are just coming now. And thank you so much. We are concluding this, and 10x will point to really conclude this Spatial Omics seminar series, because now they're accumulating and acquiring most of these technologies anyhow. So next we will have another one, the 4th one, that will be upcoming in a few weeks. I think hopefully in 2 weeks, we'll be able to finish the new schedule for you guys. And if you know anybody who's interested in presenting, please send to our way. And in the spatialomics.net, we have our emails listed, right? So you may want to contact us and help us find better new speakers. And they're always best, of course, but new speakers. And so we will be keeping you updated with this. Any questions for our end? If not, I think we will be concluding this week's and this seminar series now, and then we'll be in touch soon.
Great. Well, thank you for having me.
Thank you.
Thank you, Zach. And thank you, Ahmet.
Thank you, Rong. Bye-bye, everybody. Have a nice weekend.
Thank you. Have a good weekend.